So just before I start, I'd like to thank everyone for joining us. Put your questions in the chat. Obviously, none of this would be possible if we didn't have some sort of support, and so we're very grateful to Supernus Pharmaceuticals in Kyoto Kirin for their support of this particular spotlight today. So as you know, the way we do this is I'm going to chat to Andy. I'm going to ask him a little bit about himself, and then we're going to sort of have a little drift and a chat about genetics and Parkinson's, an area which, if we'd had this conversation 30 years ago, would have lasted approximately a minute, but has exploded in the last 25 years. And Andy's been very much at the front of that with all of his incredible work, which he's been doing. So Andy, perhaps we could start with a little bit about your background. I mean, it'd be useful to know where you began life, what you did to begin with in terms of your sort of first degree, your PhD, and then a little bit how you ended up doing what you're doing now.
Sure. Yeah. So, well, first of all, thanks for having me. It's fun to have this chat, and I really appreciate the opportunity to tell people about our work. If I just wrote this in the chat, so if I say anything that doesn't make any sense, please type and say please clarify or ask other questions, because sometimes scientists tend to talk in acronyms and assume that everybody knows what they're talking about, and that's often not the case. Half the time, I don't know what scientists are talking about. So where did I start? So I actually grew up in, I think this is what you're asking. I grew up in a small island that sits in between England and France called Guernsey. There was a book recently, people may have read, the Guernsey Potato Peel Pie and Literary Society. So it's a tiny island. There's like 60,000 people there. And I was always interested in science. From 10 or 11, I kind of got interested in science, but I was a terrible student, awful student. So I kind of didn't do very well in school, and ended up working for a year in Guernsey, and realized that working was quite hard, and maybe I should have done better at school. So that kind of prompted me to go away to university. I went to a place called the University of Sunderland in the northeast of England for my first degree. My first degree was in human physiology, applied physiology. So kind of the normal form and function of the human body. And during that degree, I got a chance to go work at a research lab, the Medical Research Council, which is the government funding body in the US for research, and the medical research. Yeah, in the UK, sorry, at the Medical Research Council Neurochemical Pathology Unit, which was in Newcastle. So I spent a year working there in a couple of diseases, Alzheimer's disease and dementia with Lewy bodies, and just loved it. I really something about research, and particularly about genetics, really just spoke to me. So went back, finished my degree, and the lab that I'd done a year out in said, why don't you come back and do a PhD? So I went back and did a PhD in Newcastle, University of Newcastle. And
what was that on, Andy? Was that? So I don't want to date you here, so I don't in terms of where was the genetics? Was your PhD on genetics, or was your PhD on sort of neurochemistry, chemicals that are wrong in the brain?
Yeah, good question. So it was from 1991 to 1995, and my PhD was mainly genetics. I actually started off at cell biology, but I was awful at cell biology. I think you probably know this, Roger. There are people that are good at it, and then there are people that suck at it. And I was in the latter half for sure. But I really enjoyed and did pretty well at genetics. So it was in genetics of Lewy body dementia and Alzheimer's disease. And this was in the days when you could look at one genetic variant, one base pair, one small change in a gene in 20 or 30 individuals a day. So, we would look at these genetic variants, and we would over time build up groups of individuals for which we knew the identity of this particular genetic variant.
And at that time, Andy, I mean, how many neurological diseases had a named genetic base? Because I know Huntington's disease, which is slightly different. I mean, that came in 93.
Yeah. Alzheimer's in
for
91.
Parkinson's wasn't until the late 90s. And the Alzheimer's must have been around then that it was starting to be discovered.
Yeah, so it was an exciting time actually for Alzheimer's, because the first mutation had been discovered in 91,
right?
And the second one in 95, second and third. So the precedents were in 95.
So yeah, to say when you started your pitch that you were right at the beginning of the age of discovery of genes related to brain diseases, and even ahead of where it first have been described in Parkinson's disease.
Yeah, although actually, as we're speaking, I realize I got the dates wrong. I'm making myself older than I appear. My first degree was 91 to 95. My PhD was 95 to 98. I'm sure everyone was surprised that it was.
I was
so
very
far
surprised,
back.
actually. I was surprised it was the last century, to be honest.
Yeah. Yeah. So actually, I was getting involved in genetics just at the time it was starting to grow in AD. In Parkinson's disease, zero. It was still thought of as a non-genetic disease at that point. Yep. Fun times.
So you did your PhD in Newcastle on genetics in dementia with Lewy bodies, where there presumably weren't many genes, and then there was an Alzheimer's disease where it was starting. And then where did you go from that? Because obviously you're now in Washington, DC, and that's a fair old way from Newcastle. So when did you move from the UK to the US, and what did you move to do?
Yeah, it's a ways for sure. So during my PhD, we wanted to do some gene sequencing. And these were the days where, now we can sequence a whole genome in a couple of days.
So the idea is that you read the entire DNA, the genes, every bit of it.
Right. So now the genome is 3 billion base pairs long. So 3,000 million base pairs long. And we can do that in a few days now. It's pretty straightforward. When I was doing my PhD, at most you could read 200 or 300 bases, right? So, and we would do this on these big old radioactive gels. So we would label the nucleotides, we would run them down this gel, and we would use the gel, we'd expose it to film, like X-ray film, and then we'd be able to read sequences.
When you say 200 or 300, would that be 200 or 300 a day, and then you'd eventually, over several years, get the whole lot, or would 200 or 300 all you could ever do on any one?
No, 200 or 300 in an experiment, really, and an experiment would take a day or two. So we'd be able to read 200 or 300 base pairs of sequence from a dozen individuals in a day, so a tiny amount. So
it'd be like, I suppose the analogy would be a bit like sort of mobile phone and cameras. When they first came on, you just got a very fuzzy picture with a few sort of blobs on it of roughly what the picture looked like, whereas now it's high resolution.
More actually, yeah, that's a good analogy, but it's probably more like in those days it was like one of those cameras where you put the cape over your back, and it's a pinhole camera versus your mobile phone now. So you began with these sort of very limited genetic studies, I would say. Yeah. So
and so when you went to the States, was that your PhD, and was that to look at Alzheimer's, or were you already into Parkinson's?
Yeah. So I was using this old technology to sequence, and there was a new automated technology, which meant you didn't have to run these radioactive gels. You could use these machines with lasers and stuff. And the lab that we knew that was doing this and had one of these machines was in the US, in John Hardy's lab at University of South Florida. So during my PhD, I went over to the US to run experiments on the samples we had collected in the UK, a series of families. So I spent a couple of weeks in John's lab. And then when I came to the end of my PhD, he offered me a job. So that's how I ended up in the US. I moved to then North Florida, to the Mayo Clinic in 99, and spent a couple of years there. And that was where I switched from dementia to Parkinson's disease.
So really, for the whole of this century, you've been looking at Parkinson's disease, but presumably, a lot of the information you get on Parkinson's disease, it's useful to compare it with Alzheimer's disease and other diseases of the brain to see if there's similarities and differences.
Yeah, extremely useful. So of course, you're looking for things like genetic overlap between different diseases, and that's maybe truer for dementia with Lewy bodies and Parkinson's disease than Alzheimer's and Parkinson's disease. But also, I think you're looking for similar mechanisms. So the point of genetics is to tell you about biology, and you can kind of infer some things across diseases. It's really key to look across diseases for sure.
Okay, so when you started your work on policy, I think it'd be useful just to sort of get your sort of view on how much it's changed. So obviously the techniques have revolutionized in the last 25 years. I mean, it's completely transformed what we can do. But I guess it would be useful for us to sort of see, what would you say have been the major discoveries on the genetics of Parkinson's disease? And I suppose it would also be helpful to hear about, because I get confused myself between things which are a gene which definitely causes Parkinson's disease. So I've got this gene, I'm going to get Parkinson's disease, versus a lot of studies where they say, well, I've looked at all these genes and it changes your risk. So I suppose it'd be useful to know, first of all, how many genes are there that actually cause Parkinson's disease? So if I have that gene, I've got a pretty good chance of getting it. And when did that start and how much has that changed?
Yeah, well, actually, the question you're asking is a very simple question, but it's one that actually most scientists, I would say, get slightly wrong. So there's really a range of genetics from mutations that if you carry them and you live long enough, you're pretty much going to get disease, through to things that just confer a small amount of risk, and everything in between. So when we generally in the field, when we talk about mutations in genes that cause disease, there are around a dozen or so genes in that space. But in truth, for some of those, they don't mean that you are always going to get disease. So one that people may have heard of a lot is LRRK2, LRRK2. So mutations you'll often read mutations in LRRK2 cause Parkinson's disease. Actually, they don't. They only cause Parkinson's disease in about 30 or 40% of the people that actually carry the mutation. So they are they're not causal, but they're a very, very strong risk factor.
Okay.
But at that end of the spectrum, where you have things that really increase your risk a great deal, there are around a dozen or so genes.
And how many people would, if you took just selected 100 people out of the clinic with Parkinson's, how many of them would sort of fall into that camp of having these 12 genes which give them a high risk, or have given them a high risk of getting it?
Yeah, that's a good question. If you around 2 or 3 in 100, however. Okay. There is a gene that kind of fits in the middle called GBA. So this encodes for a protein called glucocerebrosidase. It's much easier to say GBA. That's often called a disease-causing mutation or a mutation, but it's really a risk factor. So it kind of sits in the middle. That's fairly frequent, about 3%, something like that in the PD population, Parkinson's disease population.
So the vast majority of people who would pitch up in a clinic could have inherited some of these variations which increase their risk, but the number who have a gene which you would say, I'm pretty confident this is lying at the heart of your illness, would be relatively small. Yeah,
very small. So synuclein mutations are, as an example, right? The very first mutation discovered was in alpha-synuclein back in 1997. You're really looking at 1 in 1,000 Parkinson's patients, maybe less actually, maybe more, 1 in 5,000 would carry a mutation in that gene. So rare, but relevant in terms of understanding disease.
Yeah, so I was going to ask you about that, because obviously, if you sort of think of it in a sort of simple way, you think, well, 95 out of 100 people who pitch up on my clinic don't have any of your genes, Andy, that you're interested in.
Yeah.
So why am I interested in the 5% that have? I mean, it's obviously interesting for them, but how does that help me understand disease, given the vast majority of people don't have these particular genetic variations, mutations, whatever?
Yeah, I think it's a really good question. And I think it's a question we should ask ourselves often and early in an experimental process. I think that there's some really good evidence that at least some of those genes are relevant to typical Parkinson's disease. So let's take the very first one, alpha-synuclein. Mutations in alpha-synuclein were discovered in 1997 at NIH, and almost immediately after it was.
And that was discovered in a family, was it? That's right.
It was discovered
they found
in a family called the Contursi kindred. Yeah. So this is not a typical Parkinson's disease. Patients tend to get disease very early in their 30s or 40s. Disease progresses very rapidly and in every generation. really strong in every generation. So not typical. But the question people were asking is, okay, well, this disease looks a bit different than Parkinson's disease, and clinically, and it's super, super rare. So why is it relevant to the rest of the disease? So I think serendipity had a role to play here because, this was the first mutation that was discovered. The protein that's mutated, so you have a gene, and a gene creates a protein or encodes for a protein. The protein that was discovered, synuclein, the normal protein that we all carry and don't have a mutation, most of us don't have a mutation, that is part of the pathological hallmark that defines Parkinson's disease, the Lewy body. So it kind of strung these two worlds together, right? We have these really rare familial cases where a mutation in synuclein, in the gene alpha-synuclein, causes disease. And then every other case of Parkinson's disease, by definition, has a pathological hallmark in their brain that contains this protein that's encoded by this gene. So it was a really nice way to link them together, and I think provided a lot of evidence that studying these rare familial forms can tell us about the rest of the disease.
So I guess how do you prove that? So obviously with alpha-synuclein, you can say I found it in a family.
Yeah.
Is relevant. Well, I can stain the brain of people who died with Parkinson's, and it's there, so it's got to be linked. But all the genes which came after that, the two that you were mentioning, the GBA gene. I mean, how can you prove that they are related to getting the disease? Because you sort of say, well, they appear more commonly in people with this condition. But how do I know that's got anything to do with the disease, since it's just by chance?
Yeah, very difficult. I mean, I think in the first instance, you look for kind of circumstantial evidence. I would say, does the disease look similar? Do the patients who have a LRRK2 mutation get the same kind of pathological hallmarks that the rest of patients get? And those things, by and large, are true. As you move through the research endeavor, you start to find other clues. So we talked about this spectrum of disease risk, right? You've got these really rare causal mutations, and then you've got all of this risk down here that imparts just small amounts of risk for disease. So we know that we've identified around 90 or so genetic regions that contain variability that imparts risk for disease in just every PD patient.
Yeah.
Those variants that we find in typical PD also exert risk in people who carry a LRRK2 mutation. So they're tied again here in some mechanistic way. The same mechanism that affects risk in typical PD modulates risk in LRRK2 PD.
Okay. And so do you rely, I mean, we'll come on to discuss in a minute about, how you collect your populations and you analyze them. But I would have thought, in order to prove this, presumably you then have to go and look in cells, and you have to make animals with these sort of genetic variants or mutations and see what happens to them.
Yep.
How informative have they been? And I guess one of the things which came along 15 years ago now was the ability to make nerve cells out of patients' own skin cells or blood cells, these so-called iPS cells. I mean, how helpful has that been for you as a geneticist trying to sort of, if you like, sort of put some weight on your genetic findings that they're relevant?
Yeah, I think it's been incredibly helpful. I mean, when you say 15 years ago, that sounds like a really long time, but in research, it's actually not. It's a short time. But I think it's been really helpful. The creation of these induced pluripotent stem cells, right? Taking a patient's, like a tissue punch from a patient or a blood sample from a patient, and turning it into a nerve cell and then being able to study it. That's been really useful to understand mechanism.
Yeah.
There's also been some very kind of early understanding of disease processes that I think is kind of useful. So we talked about LRRK2 earlier. Mutations in LRRK2 were discovered in 2004 by our lab and by another lab at the same time. And immediately it was understood that this encoded a type of protein that's called a kinase, which you can think of as a switch, something that turns things on and off in a cell. And the idea was, if there are mutations in this gene that contribute to disease, and this gene encodes a protein that's a switch, maybe if we mess around with that switch, that might be a viable therapeutic avenue. And certainly, as you know, that is one of the avenues that's being chased. So I think there's lots of kind of ways to get at function and try and get at understanding that start in genetics.
So beginning about 25 years ago was the discovery of genes which were the sort of preserve of sort of families, if you like, rather rare, but suddenly became mainstream because they were linked to sort of the more common. Over time, these genes have sort of increased, and we've understood that there are more of them, and we know how to sort of investigate them. I suppose a question that comes out of this is then how do they relate to each other? So if you have 90 different bits of your DNA, which I mean, are they all talking to each other, or do I have 90 different events taking place that together give me Parkinson's disease? So how can you link what you find together and make sense of it? Because 90 seems a lot to me to try.
Yes, it is, and it's even more complicated for the risk genes because we don't know what the gene is. We have a region that contains several genes, and we don't know which gene is the culprit.
So you have a bit of DNA, and you say in this bit of DNA there are 10 things that we know it codes for. Any one of those 10, and I'm going to take a punt on one of them, but I might be.
And it gets even more complicated because, of course, we're talking about things that impart risk. They're not causal, so their effects are quite small. Yeah. So you're looking for a small effect in a region where you're not sure what the gene is that's being affected. However, there are ways to tackle this, right? We have ideas about what the function of particular genes is. So we can look across these 90 genetic regions and all of the genes within these 90 genetic regions and look for related functions. Do we see genes that are involved in one particular process or another particular process? And that is certainly starting to bear fruit.
So the idea would be you'd say, well, actually, I'm interested in how a cell gets rid of, I don't know, proteins by a process like a dustbin or a sort of autophagy. And you say, how many of these genes seem to, or these regions of DNA have genes that sort of talk to that bit of the cell? Is that the sort of?
Yeah, that's exactly right.
Okay. And so I want to just sort of, I mean, to the audience, do put your questions in the chat if you have questions for us. So as you were saying, when you moved to the States, you really couldn't do that much on the genes in terms of the number you could study. So in the last 25 years, obviously, the sequencing of DNA has vastly improved in terms of the technologies. So you say you can now sequence the entire genome, so all of the genes in a cell in a day. So how do we take this forward? How has it gone forward, this field? Because obviously in the early days, you'd have a family, you'd go searching for a gene, you find a gene, thank you very much. Whereas now you're saying I found 90 areas of DNA that are increased with the risk. I mean, how have you done that? Is that by putting together 6 families, 600 patients in population of America? I mean, how do you
great
type of experiments to find risk?
It's a great question. So as you said, when we were looking for mutations, we would rely on families. Families where a disease occurs much more often than you would expect. And this involved working with clinicians who would just obsessively track down these families, work with them to identify affected individuals. That's changed considerably. So around that era was when I moved to NIH back in 2001, and we started at NIH doing genetic analysis we had 267 patients and 267 controls in our freezer. Our last study involved genetic analysis of 1.4 million people. So,
with Parkinson's disease or
about 40,000 of them had Parkinson's disease. About 20,000 had a relative with Parkinson's disease, and then the rest were controls. So we've seen this incredible change with technology, but also an incredible change with the number of patients that we have to work with.
But just to put this in context, so if you were trying to do that experiment in 2001, on the technologies of 2000, that would have taken you the rest of your life to get mere fragments through it.
Yeah, absolutely. Well, sequencing the first genome in 2001 cost about $1.3 billion. So it would have been pretty.
And how long did it take?
Many years, close to a decade. I mean, whereas now, we can do it in a day. Now we don't sequence the whole genome of all of those 1.4 million people, but we're not far off being able to do that. I have to say, there are lots of you kind of mentioned earlier do you take the whole population of the US or do you take the whole population of the UK?
Yeah.
That's going to happen. We are all going to have our genome sequenced at some point or another. There's a truly incredible initiative in the UK, the UK Biobank initiative that includes whole genome sequencing on hundreds of thousands of individuals, not for Parkinson's disease, but just in general. And these kind of resources are growing and growing and growing.
But how would we deal with that? I mean, it's a slight side issue, but you've just told me that LRRK2, 30 to 40% of people get Parkinson's if they carry a LRRK2 mutation. So the majority of people who carry a LRRK2 mutation don't get Parkinson's. So why would I want my genes looked at to be told I've got a gene which doesn't necessarily tell me what I'm going to get? It obviously makes me worried because it now tells me I might get it, but it doesn't tell me I'll definitely get it. And given I can't do anything at the moment to stop Parkinson's, why would I want that piece of information? Surely my insurance company to know it.
Well, so I don't know the laws in the UK, but in the US, certainly you're protected against discrimination based on genetic information. Okay. But I think there's a couple of things to bear in mind. The first is what you just said. For a genetic variant, if there's nothing you can do about it, do you want to know? Yeah. The second is, I think it's a really personal opinion. So I'll say I'm biased in this regard. My wife was trained as a genetic counselor. So, her job was to give this information to people and to help them evaluate what it meant for them, what it means in terms of risk, what it means in terms of their family's risk. I think that's an intensely personal opinion. So I think it comes down to the individual, whether their view is knowledge is power or I'd rather not know for now.
Okay. So the technologies have increased, so we can now look at these vast populations, typically large populations, with great detail on our genetic variants. But I suppose one of the questions which is popped up is, so genetics tells you one thing, that gives you risk. So what turns risk into disease? Yeah, great question. So what is it that? So I guess the question would be, are there things that we're not picking up on the genetics? Are we missing things on genetics? Are there things in the environment, or what is it that's doing it? And I'll try to explain what I think, but tell me if it doesn't make sense. So we're able to estimate now what the risk is that's imparted based on genetics. So what it can say is that we think that heritability, what you inherit from your family, accounts for about 25% of the risk. So what this means is, if you were to take a whole bunch of people with Parkinson's disease and look at the average that genetics contributed in each of those people, it's about 25% of the reason they got Parkinson's disease. So it means in some people it's going to be 1%, in some people it's going to be 100%. So I'll say a couple of things about that. First of all, 25% is probably an underestimate because of the way that we kind of calculate this, we miss something. So it's going to be a bit higher, but it's not going to be 100% for sure. So what's that telling us? It's telling us that genetics is important, but it's not everything. Something else is going on. And I think that something else falls into two kind of camps. The first is environment. And by environment here, I just mean things that happen to you during life. And then the second is random, the effect of random effects, which is a bit more theoretical, I would say, and to do with a particular way that some scientists think that Parkinson's disease may occur. But that's kind of the other option. Okay. So I guess there must be certain environmental factors that I know people have talked about, pesticides, people have talked about, pollutants, well water and things like this. I mean, is there a sort of plausibility with this that these things could lead to changes in expressions of genes? So I mean, one of the things which seems to have gained a lot of interest recently has been the idea that your genes you have this epigenome, these things which sit on the DNA, which controls what genes are expressed.
Yep.
I guess the question is, do these environmental factors work through this sort of epigenome, if you like, to then cause genes to be re-expressed, or are they working completely independently and it's nothing to do with any expression of anything?
It's a great question. I don't know it's a question that we have a clear answer for. There have been some studies looking at, essentially the question you're asking is there a genetic and environment interaction in some way? So there have been some studies.
Well, also, do all environmental factors ultimately speak through genes, I suppose?
Yeah, I guess perhaps. I think it gets quite complicated, right? Because the environment is you can think of the environment in lots of different ways, right? It's not only what you're exposed to, but when you're exposed to it, how long you're exposed to it, all those kinds of things. So you can imagine that if an early exposure, early in your life course, became a risk factor for Parkinson's disease, then it must be having some kind of persistent effect.
Yeah.
So it must either initiate a process that then kind of runs away, or it must imprint in a gene that then kind of initiates that process, or it must sit in your body for a long time. So I think the idea of it plausibly affecting genetics is totally reasonable, totally reasonable.
And so a question that's come in here, Andy, which is a very good question, is about, you talk about these variants, we talk about risk, but is that different in different populations? So obviously I would imagine the vast majority of your samples come from a particular demographic in North America. And so the question is, are those exactly the same risk factors as if I was in the Far East or, in South America or something? So how much are these variants global and how much are they specific to certain population?
Really fantastic question. So the vast majority of genetic work, full stop, not just in Parkinson's disease, but full stop, has been done in people of Northern European ancestry. Right. So in Parkinson's disease, the vast majority of genetic studies have been done in folks from across Europe, actually, and from North America. Right. We even see differences within those populations. So the LRRK2 mutation that I talked about earlier, or LRRK2 that I talked about earlier, there's a particular mutation that's fairly common called G2019S. And within Northern European populations, it is, you see differences. So it's very common in folks of Ashkenazi Jewish ancestry, not so common in folks not of Ashkenazi Jewish ancestry. So that's one kind of slice of the differences. We don't know a lot about populations outside of those that have arisen from Northern European ancestry, but we're starting to find out. So I run a program called the Global Parkinson's Genetics Program. Yeah. And the idea here is to collect 150,000 persons with Parkinson's disease from around the world. So we're working with groups across South and Central America, the Caribbean, Sub-Saharan Africa, Northern Africa, Southeast Asia, all the way through Asia, really, to try and collect patients of different ancestral backgrounds and understand how is the genetics the same and how is the genetics different.
Okay, so, and there's a couple of other questions coming, which are very interesting areas. One is about if you pick up a genetic, so let's take the alpha-synuclein family, for example, or a LRRK2 family. You say, well, this family has LRRK2, but 30 to 40% get Parkinson's. Is that because that family have 3 or 4 genetic abnormalities and you've been a bit lazy and you say, well, I found a lot that will do? That's what it is. But actually, the ones who get Parkinson's have LRRK2 plus X plus Y plus Z, and those that don't just have LRRK2. So how much does one look for more than one mutation in a family? And could that explain some of the variability with some of these genes which seem to run within families?
Yeah, it's a really great question. Taking LRRK2 as an example, we know that common genetic variability that influences risk for Parkinson's disease in the general population also influences the risk for people that carry LRRK2 mutations. So we can say, that these 90 or so risk loci influence LRRK2, and that has that affects whether they get disease or not to a certain degree. There are things we're missing. My guess is that some of those will be other mutations that we just can't find because it becomes very difficult to find them in families when you have a mutation in a family that already exists. But also, one would guess, environment, exposures, lived experience.
Yeah. Okay. So then there was this is something that I was told last week, which may be not true, but it's about what your DNA codes for. So I was told last week, and you can correct me on this because I'm probably wrong, but 3% of your DNA codes for things, and 97% of it, we don't know what it does. Is that a correct statement? And B, why on earth are we carrying around 97% of our DNA, which is apparently doing nothing? Because surely it must be doing something, which links to a question someone's asked about non-coding. That there are things that are sitting there which are not obviously coding, as you were saying, DNA codes for a protein. So what are we doing about that, and what does that mean? And how much of your genetic studies pick up on the non-coding bits as well as the coding bits?
Yeah, great. Wow, great question. So yeah, it's broadly correct. It may be more like 1%. We think of as as encoding these coding areas, these things that get translated to RNA and to protein.
1%.
Yeah. So the remainder, 1 to 3, let's say.
Okay.
The remainder used to be called junk DNA. But actually, what we mean by junk is we don't know what we didn't know what it does. Therefore, we'll just ignore it for now. And we're starting to get a handle on what these kind of non-coding regions, this majority of the genome, does. Yeah. So it's really involved in regulation. So you've got these small regions that encode for a gene that results in a protein. But you need instructions as to where that protein is going to be expressed, how much of it's going to be expressed. If an insult comes along, or if a stimulation comes along, does it increase? Does it decrease? And that's really what the non-coding regions take care of. And remember, not only do you need that information, but that information needs to be different in each cell type, because our responses are different from cell type to cell type.
And at different times of our life, I would imagine as well.
Yeah.
When you're developing, presumably, it's a very different system to when you're ancient.
Yeah. And we talked a little bit about this spectrum from disease-causing mutations down to risk factors. Disease-causing mutations tend to be things that alter the gene. They alter the protein coding sequence of the gene. So they're in this 1%. Risk factors tend to be in the 99%. They tend to be things that alter expression of genes or affect where a gene is expressed, when it's expressed.
And when you do your genetic studies, are you looking at that 99%, or do you only concentrate on the 1% that are coding? So you look at the whole thing and say, there's areas here where we're getting changes in non-coding, which must have some implications for the coding bit.
Yeah, it depends on the study. So if you're looking for a rare disease-causing mutation, you're typically looking in families which are small numbers of individuals. So you really think, well, the chances are it's going to be in this 1%, this coding region, because those tend to be the really big effects. So we tend to look in those regions in families. When we're doing these really big studies, we call them genome-wide association studies. We look at the whole kit and caboodle. We look at as much of the genome as we can.
Okay. And so coming back to one of the questions, that was another question that's come in here, which is about this epigenetics. So obviously you do DNA sequencing. So you sort of say, this is what the genome looks like. And now people can sort of look at this epigenome, these bits which control the DNA outside of what you've been talking about, which are the non-coding bits of DNA. So how much can we do epigenomic sort of sequencing, so we can sort of say, this is what all the DNA looks like, and this is what it looks like on the control elements outside it?
Yeah, great question. So we can look at a fair amount now. So there's two things to say about that. What we tend to do is survey. So a lot of the genetics work we do, we don't look at the full 3 billion base pairs. We look at every 1,000th base pair, and it helps us infer information. The same is true in epigenetics. So we can look at epigenetic marks, things that alter the genome using a survey. The issue there, the limitation we have there is you have to look at a tissue that's relevant, and the most accessible tissue is blood, but is it really relevant for Parkinson's disease? So really, you'd like to look at brain tissue, and you need large amounts of samples. There are also some technological changes that are coming along, which are kind of interesting too. You talked about this revolution in genetics, where we're now able to produce a lot of data. There are some new technologies that allow you to sequence data and get that epigenetic information at the same time. So you not only get to see variation in the sequence of your genome, but also modifications to the genome. Really kind of transformative technologies.
And do you do that for representative of the whole body, or do you have to do that on individual cells, so you can actually start to plot genetic and epigenetic changes in individual cells, and presumably cells which are going to do badly in Parkinson's versus those going to do well? I mean, is that technology there?
It is yes, it is. It's kind of weird. So it's not weird, it's kind of fascinating. Science is amazing hard graft, the way you're taking one step up the mountain every day. But occasionally a new technology comes along, which is like a helicopter coming in and flying you up 1,000 feet. And one of the most recent changes is exactly what you're talking about, which is single cell sequencing. So the ability to take a chunk of tissue, chunk of brain, chunk of whatever it is that you're interested in, and sequence the DNA in an individual cell, or sequence the RNA in an individual cell, and all keep it packaged together. So what this really allows you to do is exactly the question you've asked, to say, okay, we think the disease process that we're interested in is in a particular type of neuron. We can sequence just that particular type of neuron and get information that's exquisite to that neuron. So really revolutionary kind of technologies.
So it's a great sort of contrast of scales here. You have 1.4 million people where you sequence the entire DNA. So big scale, get lots from populations, but also drilling down into individual cells and individual patients to understand what it all means at an individual cell basis. So one of the questions which come in on a slightly more global thing is about gender differences. So here we've talked about, genes, and obviously genes have something to do with your gender. So how does that factor into your studies? So how do you control for sex differences in your genetics? Or is it, it's said that, men have women in PDs about 3 to 1, so 3 to 2, so it's slightly more men than women. But how do you factor that in? And do you know anything that's told us about Parkinson's disease?
Yeah, I can say from a genetic perspective, we don't see any real consistent differences between men and women. There are certainly differences in the disease presentation and progression in men and women, but from a genetic perspective, we're not seeing anything yet. So I don't understand the difference in prevalence. Maybe it's the epigenetics that you talked about earlier, but from a genetic perspective, we're not seeing it.
Okay, and so I guess one of the questions is where is this going to take us? So if we go back to when you sort of started, we identified genes to begin with associated with families. Then we started to identify risk parts of DNA. Now do you think we've reached do you think 90 is about it? We might find 1 or 2 more, or do you think that's going to increase that inherited element, which is 25%, will grow more? Or do you think the real money now is that the genetics, we're probably not going to do anything more unless we go for the whole population, or we just go the other way, which is what you're saying, drill down into individual cells? What's your sort of vision of how genetics is going to tell us where we're going in Parkinson's going forward?
Yeah, well, I think many of the things you've spoken about, actually. So we talked about this number of 25%, the heritable component. We know the genetic basis of about 1/3 of that. So we still have a lot. And that's those 90 risk lows that we talked about. So we still have a ton left to find.
So what you mean is that there's only some of those DNA regions you actually know what the causative gene is, if you like, or
No, not even that, actually. But we have this estimate of what the heritable risk is for disease, and we know that the 90 loci, the 90 regions that we've identified so far, only account for about 1/3 of that 25%.
Oh, I see. Okay, yeah.
And it won't be 3 times 90. Once to get that full 25%, it won't be 3 times 90, because each of the remaining genes has a smaller effect than the previous one. So it will probably be thousands of genetic regions that are involved in risk in some way, shape, or form. I think that's useful to do. I think the question you were asking is, what do we do with that? What's the point, right? What's the point? So I think there's a couple of things here to think about. Traditionally, we always thought about genetics as an in to understanding disease process at the molecular level. The analogy my old boss used to say was, genetics is the opening credits to a movie. It doesn't give you the plot line of the movie, but you can tell by the opening credits, and if Arnold Schwarzenegger is the star, you can kind of take a guess as to what's going to go on. So it's kind of it's a starting point. And that's how I view genetics, as a starting point to understand biology. If we can take all of these genes that we know are involved, we can start to understand what processes those genes are involved in and look for points where these processes coalesce. They are actually the disease process, and look for, if you understand the disease process, then of course you're trying to find a space in that disease process where you can stop it or alter it or divert it. I think there's some other value for genetics too. So this idea of understanding biology is one, but I think something we've appreciated more and more over the years is that whatever therapeutic we come up with, the earlier you can apply it, the better. So the other part of genetics is being able to help predict who's going to get disease before they even know they have disease. So the first part is a therapeutic, or understanding the biology so you can come to a therapeutic. The second part is predicting who would benefit from that therapeutic. There's a third part, which is we actually think that, or many people think that Parkinson's may not just be one process, right? Everybody who gets Parkinson's may not just have the same process. Maybe it's different in different individuals. So we think genetics can help there. So you would want to come up with a biology-based therapeutic, be able to apply it to someone before they know they have disease, and match that therapeutic to the person based on the flavor of disease that they have, or the flavor of processes that are involved in there.
So you could see the future of this being that the future of Parkinson's would be treating people before they even know they've got it.
Absolutely.
And so you'd say, actually, I've got a drug. So if we come back to this LOCK2, this kinase drug, genetically, you could say, actually, I don't want to give it to everybody because most people don't have this, but I am going to take the 1% of patients who've got Parkinson's and this, and I'm going to target that because I know that's important. So it becomes much more stratified and personalized in terms of trials around genetics.
Yeah, and I think that's where medicine is headed as a whole, is around precision personalized medicine. Now, we're a ways from there. And for Parkinson's disease, genetics is not going to do all of that by itself, right? We already know genetics is not the only thing that's involved, but it will certainly be a contributor to that process and to being able to identify people before they know they have disease.
And so a question that's often asked is, people worry about, obviously, their children, their family when they have Parkinson's. What's the, they'll have heard this conversation today and said, how much risk am I going to pass on to my children? And then should I have a genetic test? Now obviously that's changed, because in the early days, if you ask for a genetic test for Parkinson's, they can only look for one gene, but now you have these panels where they can look for 100 genes. But your vision is that everyone will just have their whole genome sequenced, and we'll have all of that. So I guess the question I have for you is, what would you advise people to come to clinic saying, I want to have my genes looked at, and what's the relevance of that to my family? What does that have?
So it comes back to what we talked a little bit about earlier, Roger, which is, I think it's a very personal decision, and I would advocate speaking to a genetic counselor if you're thinking about genetic testing, because they can actually, I'm a geneticist, and I went to a genetic counselor for a disease that wasn't Parkinson's disease, and they made me think about things that I hadn't thought about. So I think it's a really personal decision. In terms of the overall risk, so it varies depending on what your genetic contribution is. But on average, a good rule of thumb is that a person with Parkinson's disease, their child is about twice as likely to get Parkinson's disease than someone in the general population, which sounds scary in the first instance. But if you think 1 in 200 people get Parkinson's disease, then that means their child has a 1 in 100 chance. Do you know what I mean? The doubling is really not much because of the size of the denominator and numerator. So yeah, unless you have a very strong family history, then generally it's fairly low risk.
Yeah, so I suppose the message would be, and it's what I say in clinic, I say I wouldn't worry about it unless you've got a better family history.
Yep.
And otherwise, you just sort of say it's a very small risk, and I wouldn't worry about it.
Correct.
So I sort of want to come right at the end here. I just want to come back where we began, really, which is one of the areas where things have been quite interesting on genetics, I would say. And again, you can correct me on this because it's not my field, but it's in psychiatry, where the understanding of psychiatric disease, where people have manic depression or schizophrenia or bad depression, it seems that the genetics is telling us that these diseases aren't quite as distinct as we once thought, that actually there's a lot of commonality across these diseases. So I suppose, given your sort of unique position in some ways, that whilst you lead this incredible global initiative on POGSIs, you've worked on Alzheimer's disease. How much are you seeing that these diseases, whilst being distinct, actually have quite a lot that's in common, or are we on safer ground to say Alzheimer's is very different from Parkinson's, is very different from frontotemporal dementia?
Yeah, good question. So I would say that psychiatric diseases are a bit different because the diagnostic criteria are kind of much looser, I would say, than what we see in neurodegenerative diseases. Maybe I would say that. Yeah. What we know generally from the genetics, though, is that Alzheimer's and Parkinson's disease are very distinct genetically. There is some overlap, but it's pretty minimal, to be honest. The disease that kind of sits in between those two, which overlaps with Alzheimer's disease, but also overlaps with Parkinson's disease, is dementia with Lewy bodies. Kind of seems like a genetic midway point between those two diseases. So there's certainly a lot of benefit to studying those three together, to look at differences and look at commonalities.
Okay. And so my final question really is that we've talked all about these genes and genetic risk and all of that, but clearly the biggest risk is age.
Yeah.
So I guess the question is why is age such an important aspect of getting diseases? Is it because as we get older, everything gets tired and so the cells can't work so well? Or is it, which it relates to a little favorite project of ours, as you get a bit older, does some of the bits of your gene which have been quiet throughout your life start becoming a bit more active because the control on the keeping your genes suppressed starts to become a bit leaky? So I mean, it's a bit of speculation, but do you think age has an effect on the genome and certain things being re-expressed or newly expressed which weren't there in the beginning, and that's why age is such an important risk factor for all diseases of the brain? Or is it just that it's everything gets tired and everything packs up and genes not really?
I think, well, first of all, I think if I could answer that question properly, I would be going to pick up a Nobel Prize at some point fairly shortly. So I don't know that I can answer it properly. My suspicion is that age is related to the amount of things that you're exposed to and the general breakdown in regulation in your biology, in your body. I mean, we're not supposed to live this long, right? We're supposed to have children and then move on. So our body is not designed to function well. Our genes are not designed to function well after a certain age. And I think that dysregulation, just an inability to deal with things that have accumulated over the years is probably a large part of it.
Okay. So you would think it's more to do with the fact that we just have more exposure to things and that everything's sort of getting a bit, because there is this theory, which is one of the theories we're sort of working on with the group, is that as you get a bit older, bits of virus which have got integrated into the genome thousands of years ago, they become a bit active. And so you start expressing these genes which code for bits of virus or look like viruses. That causes an immune response, and that starts driving disease. I mean, obviously, I don't want to put you on the spot. I mean, but I suppose it's a sort of theory about actually whether things get reactivated as you get older. And if we could understand that, and then that would offer a therapy, because we could obviously come in and stop whatever those new genes are expressing. If they're viruses, we could give antivirals possibly, or things like that.
My general view, honestly, is that it never tends to be one thing. It's usually multiple things. So actually, why not both, right? And what would be the trigger for that activation? Is it lived experience, or is it simply chronological? So I don't know that it would be one or the other.
Okay. Well, our time is up, Andy. Thank you very much for taking us through an extraordinary story, starting with the cows and the tax exiles in Guernsey, ending up running a global program on Parkinsons. I want to thank you for all the work you've done over the years on it, and how the field has transformed, and you have transformed the field. Thank you for a very educational discussion about where we've got to with the genetics of Parkinson's disease, which is a fascinating area, and it's amazing how much we now know through the exploits of you and others. So thank you very much. I'd like to thank the audience for all of their questions. It's been a fantastically interactive session. I would like to thank our sponsors, Supernus Pharmaceuticals and Kyowa Kirin, for their support of this. And that is us done for today. I think we're back in a couple of months' time, and we're taking stem cells, I think, next time. So thank you very much, Andy, and thank you very much all for joining us today.